Thank you.
Yeah, also, a minor point. This is the first time I'm doing this workshop, so there might be a few things. Okay. Um, so what we want is like AlphaFold and Rosetta Fold. It's a pipeline, and which takes a lot of decision. AI methods are, in general, anything that takes a decision, that's an AI system. That's, a smart device is an AI system. But learning methods, they need to learn from the data. And so here I want to point out this figure here, compared to machine learning and neural networks. Deep learning models are better with more and more data, and they don't need what variables are important. So for example, in machine learning, someone needs to tell them what is important, but with the deep learning, you don't need that. So when you look at the PDB data structures, and Stephen mentioned in his talk, now it started with 9 or 7 structures, and then now we have close to 185,000. And here is the some kind of timeline how homology models worked over the year. So if you look at different times, different technology contributed to protein structure prediction at different levels. The CASP came out in 1994, and we have these multiple templates that played a major role in 2002, and after that, contact maps, and then MD refinement. In 2018, deep learning models become so important. And you can see that the reason is that now we have a lot of structures in the PDB experimental structures, and also genomic sequences, right? So that's the main driver, and it was a perfect time. If somebody tried sometime here in 2008, nothing would have worked. So that's the recent thing. So not only that, in fact, the deep learning, the current AlphaFold and Rosetta Fold, they made a big jump in accuracy. They combine all of them, like the templates and contact maps and multiple sequence alignment. All the information, all the methods are integrated. Finally, there is a MD refinement, not exactly molecular dynamics, but force field refinement at the end. So everything is incorporated into the current AlphaFold and Rosetta Fold methods. Just to quickly give you an idea about the computational complexity. So this red line is the age of the universe. So right now, that's like 10 to the power of 28. I think it is seconds. And the dark line, the gray line, is the death of the universe. If you have 100 amino acids, and then if you combine all their combinations, and if you try to explore their folding landscapes with brute force method, it's not going to work. So it exceeds actually the age of the universe or the death of the universe, right? So that's where all these methods are employed. And then now if you compare to molecular dynamics and AlphaFold and Rosetta Fold models, the deep learning models, so MD would take like several days to predict the protein structure. On the other hand, if you use deep learning, so we are going to do that, like, within few hours, you can get the results. And particularly with GPUs, the prediction is so fast. So this is the diagram Sijian Wang also shown in his talk and described very clearly. So I will not go into that, but the idea is that compared to the traditional model, the deep learning models generate program as an output, and then we will supply the new input. So in our example, we are going to take the trained model that are supplied from the Google team and Rosetta Fold team, and use our sequence and then generate the prediction, which is going to be the structure, the 3D structure. So the main thing is it's going to, so we will do the whole pipeline, right? And then the first part is sequence alignment, multiple sequence alignment. And then second part, which uses structural library. So this part is CPU intensive, so it requires a lot of memory. So for practical reasons, we have to go for very short sequence and a small example. And we can, so the Google Colab will use GPU, and then Roberta Server uses the GPU. When we do the example on Amaral, we will be using only CPU, okay? But if you want to do large scale prediction, you have to use a GPU node. So, okay, so let me, yeah, so this is the idea. So if you want to know the structure, you're going to query the PDB. If the experimental structure is not known, you will go to the AlphaFold database. You know, a lot of structures are available. And then if the structure is not available in the database, you have to do the computation. You can go to the servers and submit the job. But then if you have a very large system, very large protein or complex, you have to run on your local machines or national supercomputers or cloud you have to pay. And so what we want to do is we want to walk you through these steps, but in a very slightly different way. We'll also talk about the local installation later on. Since we have the time constraint, what we will do, we will first go and submit the jobs, okay? First create the account on Roberta Server and Google Colab. You know, it's a hands-on, so we will expose the approach, how to do that. And then I will demonstrate on the Amaral cluster how to run the job. So we'll blindly submit the job, just submit, okay? And Stephen will walk you through how to query the structure on the PDB and AlphaFold database. It's important, and particularly now there is a jump when you go from, you have to extract the input ID from PDB and take it to AlphaFold DB. So once Stephen finished, we will go to and explore the results. Maybe it will take some time. In the meantime, I will show you how, when you run the AlphaFold and Rosetta Fold, how what are the important parameters you have to include in your script and relevant database and other things. Okay, and here is the GitHub repo page where I have listed all the commands and relevant information. Okay. All right. First, we will go to the Roberta server. Okay, I'm going to make it small screen. Roberta server. So I will post it in the chat. Everyone. So let's go here first. All right. And then you need to sign up for an account. I posted the link already. Maybe you have an account already. So this is the Roberta server website. In here, go and register. If you already registered, you can log in. You have to supply, your name and then email, institution, and then fill this form. It would take only a minute. I have an account, so I'm going to log in with my previous account. So let me also see the question. So if you have any questions, post them on the Q&A, and we will try to answer them as they come in, okay? So we have the support staff, I listed them on my slide, but they are here. They will help you all. And I will also check the questions time to time. So everyone able to get the login and other stuff? I think I used a dumb password. So GitHub account, yeah. Oh, I see. Thanks for the correction. Can someone post the link again? It is somewhere here. Yeah, okay. Yeah. Thank you, Anna. So what we have here is the main page. So I submitted one job, so that's showing up, right? So here you see the front page of Roberta Server, and then if you go, if you click on structure prediction, you will see submit queue examples results, and then fag and all those things. So if you go and submit, click on the submit link, click on that. And here you have a target name. So I'm going to give like, I used a Crambin example. So I'm going to use exactly the same thing. One Crambin example 2. And here you can put the sequence. So on the GitHub page, I have the inputs, then you can use this. So I would recommend to stick with this example for now so that it will finish very quickly and then we can check the results. But later on, what you can do is you can use them, use a different example, like, whatever you have. I think we don't need this. Yeah, that's the sequence. Okay. So only you need the sequence, okay? Or you can upload faster. And here you have options. Honestly, I haven't tried any of this. I know only how to run Rosetta Fold. That's my focus. This is to check, that you are not a robot, right? By the way, we will do exactly the same thing on Amaral or local cluster, run using locally installed package. Oh, Jim, is it disabled? Oh. Oh. Well, if you go to the GitHub page, if you click on the inputs, and that's where you have all the examples. Choose the 1CB, choose 1CBN.fa. All right, so, and then submit. Okay, once you cut and paste, you submit. So it will take some time. This is the job ID, and whenever the job is ready, you can check them. Right now, it says, you put the sequence, username, and then Crambin, and then length, right? And then date when I submitted, and then expiration date. So once you submit the job, it will create the results and everything in this ID. But if you don't download them, if I don't download on 23rd January 2022, it will be deleted. They can store the files for a very long time. Okay. That's Roberta Server. It's so easy, right? I mean, I really like Roberta Server more than even running on the local machine. So the only challenge is that you can do for a very large system. They have some time limit and other things. All right. So let me quickly check the chat.
Chat's clear. The Q&A is clear, Bala.
Okay. Yeah. Thank you, Stephen. So let's go to Alpha Collab. And so there are, just trying to find, so here there are two, I listed two of them. The alternative Collab is, in fact, it's much more efficient, but for the practice, we are going to do the Google provided one. which uses the reduced database, smaller database for multiple sequence alignment. But the alternative one, I recommend you to try that. It uses seek2 approach, a different method, very efficient, memory efficient. Okay, let's do the Google one. So here it is exactly the same thing. So this is the front end of the Google Colab. So you need to have a Google account to access the Google Colab. So go ahead. If you don't have a Google account, you need to create one, right? So I think most of you might have a Gmail account. The Google account here refers to Gmail and where you have Google Drive and other things. It won't take more than a minute. So please try ahead, and then I will explain here what they have in the front end. So this is the Jupyter notebook, and you can check the file and everything. The important thing, what we need to know is whatever you input in the Jupyter notebook, we can run using the run all command. All right. And so on the left side, you have, this one is the file browser. You can check the file browser on the Google Colab. It's not your Google Drive. You can integrate your Google Drive with this Colab, but we will come back to that later. So this is the icon and other things you can explore. So for the practical computation, all right, yeah, thank you, Luvan. Yeah, so that's right. It's all in the GitHub page. You can click, but I think, thank you for posting it. I forgot to post it. So now you go here, there are code, this is the comment, okay? So they write the description about Google Colab. So here, if you look at this, for best reliability, we recommend using full open source AlphaFold with the full data structure. And this one is for just for practice, all right? So here you have to just, cut and paste the sequence. You can use their sequence too. I don't know what they have, but we want to compare with what we have. So I recommend, cut this sequence and then copy the example we have here. And for multimers, you need to put multiple, all the sequences, right? We are going to do only monomer, so you can neglect all of them. And so there are, if you are interested, you can look at what they are saying here, like show code and other things. But if you don't care, just what you need to do is just insert the sequence here, and then when you go to the runtime, click on the runtime, and then say run all. Okay, so that's going to run everything, and then say yeah, it's okay, and run. So see this circle that is rotating, that means this part of the code is, in all of them are executing, and then here it shows, okay, so this is a step it is running right now, only 0% complete, and we will come back to this later, okay. Quickly, if you look at here, runtime, and then change runtime type, we are not going to change, I'm going to show you. So right now we are using GPU. So Google Colab provides free GPU node for you, but the restriction is you can run up to 2 jobs, and I think maximum of 12 hours or something execution time. So I'm going to cancel that, okay. So that's Google Colab. So this is going to run for some time, and we will come back and check the results, all right. So for now, what we'll do, we'll do the final job submission, which is Emerald. That is going to be only for those who have account on Amrel. I sent an email to all of you asking to apply for an account if you didn't have. So I hope those Rutgers participants will have the accounts. If you don't, if you are from outside, that's okay. You can watch how this is done and take the scripts, modify it to your local cluster. That's the best thing, right? So now, yeah, let me quickly point out how we can access Rutgers. If you're outside Rutgers, you need to go to the, you need to establish a VPN connection. So let me post it. Oh my God, why is that? Yeah, I'm trying to copy and paste. So what we want, if you're outside Rutgers, Jim says it's too fast. Okay. Yeah, okay. Sure, Jim, I'll do that. Very quick, yeah. So what you need is once you go to the Google Collab, and all these stuff are kind of predefined. The only place you need is to insert the sequence, the FASTA file, which you get from here. I can also copy and paste. Copy, paste, and then in the Collab, and then here on the runtime, click on the runtime, and then say run all, and it should start executing everything. One possible thing might happen is I might have done it long time back. It might ask you to whether you want to add Google Drive integrate Google Drive with your Collab environment. If it says, say, go ahead and then add it or cancel it, whatever you like, that's okay. But as far as the job is concerned, you insert the sequence here and click on the runtime and say run all. That's it. It should be running. I hope it's okay, right? Yeah. All right. Now, okay, great. Yeah, so this is the major part. So why do we want to run on Amaral or any cluster? No, because they provide much more resources than what you get freely from Roberta Server or AlphaFold Collab. If you want to run big sequences and big protein or multimers, for example, spike protein, it's so hard to run on Roberta Server. So you have to run on your local cluster, all right? For Amaral, we provide two types of access. One is secured shell, another is web. If you want to do web access, you can go to open on demand, right? And then, yes, yeah, you can go there and then give your net ID and password. You will be in. I'm going to show you that. So you see it's asking for net ID and password. Now I log in. Now I have saved it, so I'm going in. Sometimes it may ask for restricted access or something. You say it's okay. No. It will say it is not secured. You say it's okay to go. So Samir, I think you need to you need to have a net ID and you need to establish a VPN connection. Okay. So once you go log into open on demand, this is the front end. So you can see here, and then click on Amaral cluster shell access, and then again you have to give your password. Well, I typed my password incorrectly. So now I am in, right? So okay, Kenneth says you're in. Good, good. That's one approach, and there are other ways to access Amaral. Yeah, and then if you install a Chrome secured app, that will do exactly the same thing. So you can download this web app, freely available, and that should, once you launch that app, it will allow you to run the secured shell just within your browser itself. So I'm saying that because some of you may not, for example, Windows, right? Windows user, they may not have a terminal like Mac or Linux. So if you're on Mac or Linux, you can open a terminal. Secure shell is kind of already installed. You need to just do SSH on a terminal. You do SSH and netid @amaral hpc rutgers.edu. Okay, so that should take you in. For Windows, either you use Chrome shell or Git bash. You can install Git bash. I put the link here. That should give you, once you start that has the secured shell application integrated. All right, so you can do that, and then do SSL, the above command. But if you don't have anything, if you don't want to install anything, go with Open on Demand or with FastX. Okay, so you can use either SGPS. This is the FastX application. You can use that too. Okay, so these are the ways you can get into the cluster. All right, once you are inside the cluster, what you need to do is, now we are going to run the example. So we used Roberta Server to cut and paste the sequence and run the job, and we did that for Collab. Now we are going to do something very similar for Amaral. So on Amaral, everything is based on Slurm scheduler. That takes a long time to explain. Some of you know already. The advantage is that you can run long-running jobs, very powerful jobs that gives you more power, more storage. Right? So we have installed the package here of a fold and Rosetta fold. All you need to do is, come up with your sequence and run your jobs. So we are going to get the examples here. So I'm going to use my Scratch space. It's up to you what you want to use. Let me make the font bigger. I think this is better. Okay, I think. Yeah. So if I have a directory called workshop. Yeah, empty. Good. So there are in two locations I have the example files. One thing you can do is, you can do Git Clone, and then where is my directory? Yeah, here. Oh. Peter, yeah, that's a good idea. I don't know. We should have done it while we started the Zoom chat. I don't know. It can be done now. Steve Wu might want to give a try. Otherwise, I know an exact idea. So maybe Vlad or Janet, Paul, or or support staff can copy the important commands on the Google Doc, and then they can share it here. That would be another option, a quick option.
Peter, did you try to press on these three dots at the back, at the side of the chat? When you hover a mouse, a cursor, the three dots at the chat, and then click on it? Yeah, thank you, Ken.
Joey says you don't have, he doesn't have any problem. My 3 dots. Okay, so I leave it to work staff to sort this out one way or other. Or you could cut and paste on the Google Doc also. Okay, great. So now we want to quickly run the example on Umbrella cluster, right? What I want to do is I want to copy Git clone the whole directory. Where am I? Yeah, here. It's easy to get lost. Oh, I can do copy now. Now I understand what's the problem. Okay, so the open on demand is not letting me to copy. Oh, that's so bad. Maybe copy and paste. No, it doesn't help. Control Shift V, Control Shift V. Oh yeah, thanks Joey. For open on demand, to copy that, use Control Shift V, right? Here is the command, clone, Control C, paste. Once you do that Git clone, you will see a directory, AI fold tutorials, and then, oh, sorry, Peter. I think, one of the staff member can copy and paste the relevant commands in Google Doc, and then share it with the community. So, okay, so inside this, we have everything. What I would recommend is to run this command. So everyone able to copy the files? Oh, I put that in everyone's one. Okay, I need to send to everyone. Yeah, okay. Let me see. New Google Doc, and then paste, share. Oh, okay. A workshop. Of course, save. What is that called? Oh, share with.
Recommended to change everyone who has the link instead of restricted.
Yeah, change everyone with the link, right? Correct. Yeah, thank you. And then that's the one I was checking. Editor, copy link, done. And okay. So this is the link where I will post. Maybe from here you can copy and paste. So whatever commands you want, you can ask the staff to copy here, and then you can copy from here, back and forth. Yeah, sorry about that. I should have thought about this before. I didn't realize the chat box would not allow you to copy. So okay, so once you copy everything, right? So in this directory, we have subdirectories. AF2 is where we will do the examples of AlphaFold 2 subdirectories. And then there is a finished job. We will take a look at that later on. And then RF is where we have example for 1CBN. And then under AF2, we have 3 different directories with 3 different options. One is full database, another is reduced database, which will finish a little faster. And then the third option is use MSAS. That's an interesting one because if you have really large protein and it demands more memory, so you run the first part on the CPU and finish the multiple sequence alignment, and then take that and then run the GPU part separate. So here we are not using GPUs, but we are going to pretend like, okay, somebody did that on a different large memory machine and got the sequence alignment, and then we'll do the GPUs part second. So those are the things. All of them, we are going to run it with a single command. So type chmod +x run all jobs, okay? So Kenneth says permission denied. Oh. So what you did, Kenneth? I don't know, you are one step ahead of me. Run all jobs, okay. Right. CP target. I don't know where you try to copy in a fold directory. Oh, in that directory. Yeah, right. So you cannot, yeah, okay. That's another, yeah, I'm going to show you that. So this is one way to copy from GitHub and then, create that run all job script ready to submit. Yeah, you need to change to, okay, that's a good point. So Jim pointed out that once you clone and then you go cd control V, the cd command would be small c, and then do the chmod +x the command. So all these commands are also kind of listed in the GitHub page. If you go down here, so this is how you SSL, you do git clone and then change here. And then there is a typo, I think, because someone pointed out that, so you have to be careful here. And then here you need to do chmod +x, then the next step. So I will do the other approach, which is copying. So this is one way to clone from the repo. Other thing is we have the files, projects, org, training, users, training, and then AI fold. And this is the one we want to copy. Okay, so -R, Control-Shift-V. Yeah, I'm going to copy like AI for fold because I have got another one. You see, I have that. So, and then all the files are exactly the same inside, right? So what I can do is, let me go back to my tutorials, right? And then now you just run this command, run all jobs. And that should give you like, submitted job, 169, all those things. And so this comes after this part. So once you change this, you do this, paste, right? Yeah, that is a good question. Yes, how it was installed. That part I'm going to walk you through after Steven finished his PDB part. So now we submit a job. I hope all of you submitted. I will give you a minute or two for you to submit the jobs. And then Steven will walk us through the query part. That's a very important thing, right? So everyone is okay, right? Anyone able to submit the jobs? Yeah, Joby says that, okay. So I can quickly check how many jobs are running. Jacques says no, okay. So yeah, then once you submit, like Joey said, you have SQ minus space minus U and your net ID. That will, oh, what happened to my job? Oops. Oh, maybe that mine appear to have died too. Okay. How about the one on here? Okay. So let me quickly debug this. Yes, that's we are going to walk through the entire slum file. Control show job. Ah, 16971820. So let's see. I might have made some mistake in the job submission script. Is it running first of all? Yeah. Yeah, okay. I would recommend, so what happened was this, okay, so I recommend copying this folder then. Where is that? Yeah. Yeah, please try this. I might not have changed the reservation name. Oh. This is so tricky. Paste. Yeah, this is what we want. Oh, it goes directly to Joy. Okay, so anyone else able to submit the job? So notice the dot at the end. Okay, space and dot, and that's what you need. Let me check quickly. Yeah, SQ. So yeah, there are two things. One is that you should have got that. I see some jobs are running, maybe only mine. So the thing is that, yeah, control show reservation. I added most of them, IQB, AI. So this is the reservation. So if you are one of this person, you have been added. Oh, you are there? Okay. Try this. And then you should be in there. And then, so the script should submit a job to everyone. So when you copy this file using cp command, so my jobs are running. Oh, okay. Is it using the reservation? Grep -I reservation. So yeah, it is using the reservation. Everything is fine. So okay, Kenneth, I will post your net ID to me, and I will check out why that job is giving you error. So if you know, then let's let Stephen to run through the PDB query search, and I will check why it doesn't work. And then we can, anyway, we have the Roberta server and other things coming up. Stephen,
I'm ready. Thank you very much.
Okay.
I've been listening intently, and
Yeah.
I will share my screen now. And so we're going to do, as Bala indicated, we're going to do three things. We'll start with the PDB, and I'll show you how to find the PDB structure of the protein that I talked about during my earlier presentation, and then we'll jump to the AlphaFold case so that you can get practice in getting access to any one of the currently 350,000 computed structure models which are stored at AlphaFold DB. So if you, I'm going to just provide you with the guidance and the screenshots. You can go on the web with your browser to rcsb.org, and if you type in Nova-2 in the top search box, a drop-down menu will appear, and you will see an opportunity to click on crystal structure of Nova-2 KH3 K-homology RNA binding domain. And so if you do that, you will then be taken to the results page. You have to scroll down a little bit because there's some information about the search, and when you do, you'll find that there are two structures emerged in this related to that particular topic are present in
Stephen,
the search.
We are not seeing your screen.
I'm sorry, I neglected to share. You're absolutely right. So let me share first. Sorry. So let me go back. Sorry. Okay. So if you go to rcsb.org and then you type Nova-2 into the top search box, as shown here on the screenshot below, a drop-down menu will appear for various elaborations on that search. And if you click on the first one here, crystal structure of Nova-2 KH3 binding domain, you will then be taken to a results page. You have to scroll down past the some verbiage pertaining to the search itself to get to the results. And the first result that you should see there is protein data bank ID 1 Delta Tango Juliet. This is the structure of the RNA binding domain by itself, no RNA present. So if you click on that, you will then be taken to what's called the structure summary page for 1 Delta Tango Juliet. And then you have the opportunity of, with a single mouse click, of getting access to a 3D view of this protein structure. And that's this particular button here that's shown within the red ellipse. So if you were to push on that structure, you will then be taken to the Molestar viewing window. Molestar is a molecular graphic system that's being developed jointly by the RCSB Protein Data Bank in the United States and Protein Data Bank in Europe, which is located just outside Cambridge in England. This is an open source software project that many people outside the two Protein Data Bank data centers are actually contributing to. And you can, by placing your mouse, your cursor over the screen here, you can actually manipulate this image. You can make it larger, you can make it smaller. Documentation is available on our website, rcsb.org, to teach you how to use Molestar. The beauty of Molestar is that it works in your browser. No external software is required, and it carries with it all of the information that is in the PDB files for this structure going beyond the atomic coordinates. You can also, if you look for, you can do structure similarity searches or sequence similarity searches from this page, from the structure summary page, and in doing so, you can make comparisons with other KH domain structures in the PDB, and you'll see that they're very similar in 3 dimensions. This is a compact alpha beta domain that is specialized in the role of RNA binding. So this is a way of getting access to the PDB structure information for the Nova-2 protein in isolation without RNA bound. So are there any questions or concerns? I'm not able to view the Q&A or the chat. So Bala?
No questions.
Okay.
So far,
Okay.
Stephen.
So hearing
maybe
none,
people are thinking. Yeah.
Yeah. So hearing none. So what you've been able to do is to get information about a portion, only a small segment of a much larger polypeptide chain, and you may want to visualize next the AlphaFold computed structure model of the full-length human Nova-2 RNA binding protein. So for that, what you need to do is to go to the alphafold.ebi.ac.uk site. This is the AlphaFold DB site that's maintained at the European Bioinformatics Institute. And there's a search box that you need to type the UniProt identifier for this protein into, and the UniProt identifier is Quebec 9 Uniform November Whiskey 9. So if you type that into the AlphaFold DB structured database search box and click search, you will be taken to the results page for this particular UniProt ID. And at this point, they want you to confirm that you've found the right protein. So you can see in the red ellipse is RNA binding protein Nova-2. If you click here, to confirm that is in fact the protein that you're interested in, then you'll be taken to the AlphaFold DB page for that particular prediction. For the Nova-2 protein, and you can scroll down to their 3D viewing window, which also works using the Molestar technology. And by placing your cursor, your mouse into the viewing window here, you can manipulate this to produce views like this. And I want to just stop here for a moment and remind you that the blue shaded parts of the structure are very well predicted. So you can see the third KH domain here. That was the KH domain that you were just looking at from the PDB structure. And then superimposed on top of one another is our KH1 and KH2. There are 3 KH domains in this protein. 2 of them actually will form in the crystal form a complex. And that structure has also been determined, but in the context of the Nova-1, very closely related Nova-1 protein. And then if you look elsewhere in the polypeptide chain, and this is very important, what you see are the yellow and red sections, and these are the very low confidence regions. You cannot trust the atomic coordinates for these parts of the predictions. They are simply not reliable. And you should not be using the structural information that is downloadable from AlphaFold DB for these parts of the structure. This part of the structure is very accurately predicted, as are the other 2 blue globular domains shown here. The rest of it is not well predicted. So you can think of this in 2 ways. I will ignore anything that is low or very low confidence because it's not reliable, and focus my experimental studies and my planning of future experiments on only the parts of the AlphaFold computed structure model that are predicted either with confidence or with very high confidence. You can think of it that way. Alternatively, you can think of the portions of the polypeptide chain that are not confidently predicted by the AlphaFold algorithm as parts of the structure that you might want to focus on using other experimental techniques. It's clear that given what we know about the relationship between these very low confidence regions and intrinsically disordered segments of proteins, these probably don't have a defined structure when the protein is in isolation. So you may want to do experiments using other techniques to probe what the function of this portion of the protein is and these regions that are not well predicted. So questions or concerns?
So far, no. Oh, there is one, but that was for me, I think. Yeah. Yeah, I don't see any question, Stephen. So, Stephen, there is a question from Joey. I think Joey, can you download the structures with the low confidence regions omitted?
Bala, I think Stephen had a connection problem. And
okay. So yeah, we will go ahead. Yeah, thank you. I thought, yeah. Oh, okay, yeah. All right. So what we will do, we will continue with. Yeah, screen is frozen. Okay. Yeah, that happens. You know, that happens a lot to me. That's why I try to connect with two sometimes. All right. So what we will do, I will get back to the question shortly. What I want to do is, finish up the thing, like, I don't know if anyone's job finished. So mine is still running on AlphaFold. You see here, it's using this much RAM, this much disk space, and certain steps are finished, like the installation steps and making prediction. And then it is still doing the multiple sequence alignment. That's very expensive in terms of memory. Then only we will have the results. Okay. And let's take a look at, I think even Roberta Server, it is still running. Okay. So what I will do is I will quickly show what I did before so that you will get an idea of what it is like, so this is the job that's finished. Where is it?
Bala, well, we can't see your screen. I don't know if
sorry.
you forgot
Thank
or
you.
it wasn't intentional.
Thank you, Udi. No, I did not share the screen. Forgot to do that. My job did not converge either for AlphaFold or Roberta Server. So I'm going to check what we have before. Like, this is an example result. You will get something like that once it is converged. So this is for a different protein. Still, the example is running. So you will get a structure like model 1, 2, 3, 4, and then error in terms of position of the residues. You can visualize, where, like what Stephen mentioned, the loop area. I should have done exactly the same protein, but this one is different. So wherever the error is high, you see that that's more like a loop area, right? Loop region. So that's more what you will do with structural biology results. So you can then, in terms of job, I think you can download it. Once you have done, you can download. all coordinates less than 5, everything?
Bala, Stephen, I'm very sorry. I had a catastrophic failure of my computer.
Okay.
And that's why I suddenly went dark.
Yeah.
So
I will continue.
you continue. I will answer questions. I had come
Yeah.
to the end. I will answer any questions in the QA.
Yeah. Okay.
In
There is
writing.
one in the
Apologies.
chat for
Yeah.
you. Yeah.
Okay, I will find it. Thank you.
Yeah. Okay. So this is where you can get results on Roberta Server, and then same with AlphaFold. It's still running. Let me see if I have my previous results. It's only the notebook. Let me see if this got the results we want. This is the same one. Yeah, this also doesn't have the results. I thought it would finish. For me, it finished very quickly since it is GPU. On GPUs, they are supposed to finish like within 30 minutes. Unfortunately, they did not finish yet. Okay, so let's go back. Yeah, let it converge. I will explain the code blocks in the job submission and then come back to this. All right, I see that some of your jobs are running, so that's good. If you go to Scratch, let me make it bigger. CD Scratch 387 workshop. I think workshop and AI fold, and then AF2. Let me go through one of the example here. Maybe I can go to the finished job. So once your job would finish, you would see the output, output, and then inside input, you should see the PDB files like this, 5 models. And pickle file is where it stores condensed format, all the information about predicted values. We will take a quick look at it. Would order something like this, ranking debug, and this is the global score, and which means the model 5 is the best model out of these 5 models, predicted models. And so there is an analysis script which you could run. So let me copy that. Yeah, I'm going to copy everything. Analysis script. And if I extract, this is basically, I'm getting the same thing what Stephen showed in his AlphaDB output, where the loops, certain parts are flexible, they have low confidence region, and so we want to extract that from the data. So basically, what each model does, the predicted model, we know that, for example, ranked 4.pdb, that's the best model, right? And then if I look at that, It has the atom information, X, Y, Z coordinate, and then the last column, one before the last column, has the predicted TM score. So we can visualize that. Let me also run the loop RMSD, which is basically a script that runs through all the predictor structure, and that will give you the results. So you can run the same thing, but I'm going to do, yeah. So this, either I need to transfer the file to my local machine to visualize that, or I can go there and then check the output using the graphical interface, right? Workshop. Finished jobs, and then the output and the input. This is where we will have. Do we have that? Yeah, I have that. Firefox. Seeing. I don't know why it is not. It should do the tab completion. So this is the predicted, this is the residue ID, and then predicted TM score. You see that there are some loops which doesn't have a very good score. So those having more than 90, they are good regions. That's what Stephen has shown for a different protein. That's one thing. And the other thing is, yeah, you can look at the RMSD score. So compared to the experimental structure, the predictor structure has an RMSD score of 0.56. Okay, so really very good, right? But this is a small protein. So that's good. So that's the output. So you have to analyze the relaxed structure and then the scores. If you want more detail, like PAE score, they are embedded inside that pickle file. So I will not go into the detail, but I will focus now on how you can submit jobs with the different options. What is the jobs of submission script looks like, right? So in the Jupyter notebook and Roberta server, everything is hidden. And I try to hide that through a script, but when you do actual computation on the cluster, you need to have some understanding what database is being used, and the reference database, like for genome database and UniProt database, and where they are located, and what binaries are involved, and what options you can do. So the main script that we submitted is the slum script. So I kind of simplified it. So what we are doing here, this is the slum standard thing. If you don't know about this, don't worry about it. So we have separate workshop for that. I recommend you to attend that. So I'm running, this is the wrapper script. It does one thing. Okay, so it takes in the input FASTA file from the current work directory and creates the outputs in the /output. And this is other script, like, I run part of the analysis within the same script, but this is the AlphaFold job. Now, let me take a look at the AlphaFold job. What we have, so these are the environmental variables you have to define. So this is for GPU, and I defined it. It doesn't hurt, so I defined it, but you might want to drop it for CPU jobs. So first you have to define where the database is located. So we have copied the database, which is around 2 terabytes in this location. And the binary directory is located here in this location. So in here, the binary, so AlphaFold uses a multiple sequence alignment using jackhammer, HH-blitz, and hidden, HH search, and k-align. All these binaries are required. to run that multiple sequence alignment. And also it requires, where the template is stored, and that's where we define database directory and then point it here. But you can also directly say where the database is located. Okay, so we have installed this already in that particular location, and you have to do is define it so that AlphaFold knows where the database is located, and where is that? Yeah, where is database is located and where is binary is located. Now, these are like, additional things we need. We are loading GCC and OpenMP modules, but these main two things are, the AlphaFold path is set using these two commands, load AI fold and then activate AF2. So that's subdirectory there. Environment, Conda environment. So we are using the Conda installed AlphaFold 2. You can also install, we also have Singularity image. That's another approach. I preferred this one because I can easily add some additional tools. For example, I'm using PyMol. Suddenly I may change and then add some other tools like PDB tools. I can add time to time. So I prefer the Conda installation approach, but the Docker or Singularity approach is another one. You can do that too. Personally, I prefer the Conda installation. And if you want to if you want to know more detail about how the installation, how we, what are the things we do for installation. So I have an installation directory within the GitHub repo. You can take a look about the basic stuff. So the installation is not a big challenge, but definitely the database, it's like 2 terabytes. And if you want to add multimers, you also have to create that, and that's a bit of challenge. The package itself, either Conda or Docker, that's okay. So these are the general options about database directory and binary directory. And the main thing, the options you have for monomer, you have to use monomer. So I'm not going to cover multimer, so we'll stick with monomer. Database, you can use either full DBs or reduced database. Okay, so there is an option for that. And then the template, I'm using the most recent template. But if you want to exclude anything, so say for example, you don't want to include the most recent PDBs, and then because you want to do some competition, then you have to use the templates before certain date. And the other thing, yeah, this is the main job. Okay, now let me show you the difference between the reduced database, or maybe the use MSAS. Suppose, I did the multiple sequence alignment before, then I should have a directory here with the pick. Oh, I should remove this. The feature.pickle file is required, and also whatever files inside the multiple sequence alignment, they are required. Then what I can do is in my submission script. So here I'm using a different submission script. Submission script is same, like, run CPU, and I'm running the wrapper script, this. And then the main difference with this wrapper script is I'm using use pre-computed multiple sequence aligned sequences, right? So that's the only thing. Then this will run in station. The main advantage with this approach, you do the CPU intensive part, the memory intensive part first, and then do the GPU next. The reduced DBs, they are like, everything is going to be the same. Few things are going, no, there are a few differences. The main difference is that the database path, where is that? Yeah, here. I am using, This is incorrect. I should be using reduced data. Oh no, actually I'm using it here, but then maybe here I am redefining. Oh, this is not correct. Yeah. Yeah, this uses the full database example. Yeah, this is a full database. Okay, yeah, sorry about that. So basically you have to define, instead of full database, you have to use reduced database and give the respective directories, define the respective directories. Now, community of a fold. So I think I will take there is only 5 minutes. I just show you the size of, we added some extra things. So approximately you see that it's like, around 3 terabytes of space. We have some duplicates, but that's the size. And the models,
Is
where are the models? Is that it's hanging. So what is it, Paul? Oh, okay. So I think, so we learned that, Sijin also said, deep learning models generate the programs or the rules. And that's what is, maybe I can use this. Oh, I too have network problem in my shared screen. I think with Emerald cluster, but so anyone got the results? Yeah, okay. I think I have got some results. See here. Oh, still it's running. I think we are very close. Still it is running. But at the end, you will have whatever I have shown you through the PDB, something like this. You will have all the results and certain metrics, the scores. How good is the result? So anyone got results? Anyone? Let me see. Maybe in the chat there is a message. Same status. Oh, okay. Yeah, GPUs. Run AlphaFold shell is part of the AlphaFold 2 installation. Yeah, certain, we don't, I didn't change any of the scripts except for multimers, where I have to shut off the relax, but we are not discussing it here. So there is nothing. So let me answer the question here, and then maybe in a few minutes it will converge. So we have 8 more minutes. I will try to finish in 2 more minutes. Oh, that's the one. Yeah, we need to extract it from the pickle file. I have not included the script here. Contact me. I will send you the script. So all the information about PAE are stored inside that pickle file. For example, if you look at ranked features.pkl file, and it should be there. You can try that. It's easy. Yeah. So, okay, I think, it is okay. The idea is not to, when you go home and then if you keep this notebook open, you can see the results when it is finished. All right. Same with Roberta Server. Maybe Roberta should be fast. My queue. Yeah, this one is one completed, the same thing. So same thing like model 1, 2, 3, 4, 5, and here you have the metrics here and the confidence, right, for different models. This one is 0.91 and model 1, oops, yeah, I think it's the same 0.91.
This is a very easy case, so we would expect that the algorithm generated a good result. And if you just scroll down a hair, please, so you can see that they're estimating the atomic coordinate error as being about 0.6 angstroms for most residues, and then there's a loop region that is not as well defined, but even a predicted error of 1.5 angstroms is still still quite usable in terms of planning an experiment with a computed structure model. So this is, I think that this is a success, and it shows the enormous power of the technology, whether you're using Rosetta Fold or the AlphaFold, or downloading the data from AlphaFold DB. I should have mentioned earlier that the models of the yeast binary complexes that Minkyung Baek was talking about earlier are actually all available from the Model Archive, which is maintained by the Swiss Institute for Bioinformatics. And you will see during the course of the next weeks to months that the Model Archive will grow dramatically with both Rosetta Fold and AlphaFold predicted structures coming from teams other than the Baker Lab and teams other than the DeepMind group that's located in the UK. So, Bala, I suggest that we move to the acknowledgements.
Yeah, sure.
Yeah,
Just
okay.
one point. Yeah, you can here is the download link, you can download the models. Same for Google Collab, and then Emerald results will be on Emerald. That's it. Yeah, thank you.
Terrific. So if you could stop sharing your screen, I'll share mine, and you'll do the honors with the last couple of slides.
Yeah.
Okay.
Yeah.
Over to you, Bala.
Yeah. Thank you, Stephen. It was really great pleasure to run this workshop, and many people contributed. We like to thank the speakers, all the participants who were sticking around for a long time till the end, or many came and gave their knowledgeable input. And we would like to thank IQB and RCSB PDB group, Maria, Michelle, Steve, Sagar Khare's lab at IQB, Joseph, Joey, he did a lot of help. And in fact, Sagar and Joey were the first one gave me the feedback when I ran the examples. And then Office for Research at Rutgers, Mike Zwick, who gave the welcome address. Rutgers work staff, they have been helpful from the beginning to run the examples and other things, and answered a lot of questions throughout the workshop. Paul, Janet, Vlad, Woody, RCSB PDB funders, NSF, NIH, DOE, RCINJ, and we would like to thank also PDB 50 sponsors. Thank you all.
And those sponsors are shown on the on this last slide. So thank you, everybody. Thank you, Bala, for your leadership, and thank you, everybody, for participating. If you have any feedback for me or Bala, please don't hesitate to provide it. I'm going to put my email in the chat. All feedback's good, particularly negative feedback where we could have done better. So thank you very much. Good night, everyone.
Good night.
Yeah.